could only pump 6 because they skipped complex 1, complex 2, not a complex. They will also see in the next lecture when we talk about bringing lipids into the picture, that lipids as well are capable of oxidizing electrons and also feeding them into the Q pool. Again, those lipid electrons, when they move through this pathway, are also going to bypass complex 1. And so once again, fewer protons. So not all electrons, right, are worth the same amount. NADH electrons pump 10 protons. Electrons that were extracted onto FAD to make FADH2 only worth 6 protons. Anyway, here we are. This is kind of what we've done so far, right? We've gone complex 1, 3, 4, and 2, 3, 4. We've pumped protons. So what's been the point of all this, right? So what do we have? What has this done for us? What this done for us is really to create 2 different types of gradient, which collectively we know as a chemiosmotic gradient. We have created a gradient in terms of numbers. We have more protons on one side of the membrane than we have on the opposite side of the membrane. We know that that is a situation that is out of equilibrium. It took energy to create it. That means that we have energy now stored up into this gradient. But we also have another type of gradient that we should keep in mind, which is that we have a charge gradient as well. We have more positive charges on one side of the membrane and more negative charges on the opposite side of the membrane. Both of those things represent stored energy. Both of those things are the energy that we now have. In having more protons on one side of the membrane than the other, we have a chemical gradient in the form of a charge. We have an osmotic gradient in the form of the fact that there's a disproportionate number of protons or molecules on one side of the membrane than the other. We have to use that energy. And it turns out that we can use that energy because there is really only one way back into the membrane here to restore the cycle, and that is through an enzyme complex known as the ATP synthase. And so somehow this enzyme is going to use all of this tremendous amount of energy that we've stored up into the gradient to make ATP, and probably not in the way you would think it would. Just to remind you, here's the gradient we have, right? So we're on the positive side of the membrane. This is in the cytosolic side between those 2 membranes. We have lower, we pump the protons out here. Here's the higher pH. This is what is called the proton motive force equation. So there's some practice problems for doing so. It can be expressed either in the form that we saw last semester here, where we have the natural logarithm of the ratios. But since we're only dealing with protons, you also can express that just in terms of the differences in the pH. You'll note that the Z here disappears, but that's because we're talking about protons, so it's actually only 1. I don't know, sample calculation here, I don't know, we have to work through it all together. It just gives you some sort of information into how much energy is stored in a typical gradient. This is using this form here, but I think you should be comfortable doing problems with both forms, right? Either the natural logarithm or converting it to the form which uses the regular logarithm. Anyway, how do we know that this proton gradient is so important? This is kind of something cool and also really interesting, is that there's a class of molecules that are capable of stopping the synthesis of ATP that themselves do not interact with the ATP synthase. In fact, this was really pivotal in understanding how the whole mechanism works, so we should take a look at these molecules. They are collectively known as uncouplers. And what uncouplers are basically molecules that are capable of gaining or losing protons and drastically changing their solubility properties as a consequence of that. So you can see, for example, where you have this molecule here, 2,4-dinitrophenol, is here, but it can pick up a proton where there's an excess of protons. When it does so, you'll recall from biochemistry 1 that if we have a group that's charged, that's now just modestly polar. If everything else here is relatively nonpolar, it can actually pass through the membranes. Since there are fewer protons here, the Chatelier's principle says that this proton will be dropped off. And if you treat mitochondrial membranes with these uncouplers, you actually result in a loss of the ability to produce ATP. And so that was really the evidence that the proton gradient was directly coupled to the synthesis of ATP. But of course, the origin of that is even cooler, because nobody was looking for a proton gradient in the first place. It turns out that this DNP, the 2,4-DNP, was discovered inadvertently, or this phenomenon was discovered accidentally, by people who were working in explosives factories, because DNP is used for the production of explosives, and these people had a tendency to do 2 things that were quite pivotal. One is sweat profusely at work, even when it was not very hot, and 2, lose massive amounts of weight. I mean, these people became extremely skinny. This turns out to be because they were actually inhaling this stuff, and this actually led to the first ever diet drug that was ever introduced on the market. DNP was actually, 2,4-DNP was actually sold as a diet drug. People took it, they lost a lot of weight, and then unfortunately, exactly as what you might predict, they lost a lot of weight because they died. Yeah, it just killed pretty much everybody who ever took it, and so it was actually banned. One of the first things that was ever actually banned as a pharmaceutical, and it was gone throughout history, only to be found in biochemistry slides and talks about the electron transport complex, until it was brought back to life by the internet. Now, yeah, you can actually go online and buy this stuff. And also, not surprisingly, people definitely lose weight if they take this, 100%. And also, people die from it again. So it's just another amazing thing that the internet has managed to bring back to life. So like don't, okay? I mean, this was the first diet drug, but if you take even a slightly higher than normal amount of dose of this, you will basically sweat yourself to death. Anyway, here is the enzyme that we're talking about. This is the enzyme that we are going to use that proton gradient to make ATP. This is also an enzyme that I told you about in biochemistry 1, a mystery enzyme that I guess the suspense has still dissipated. So let's talk about this enzyme. First of all, this enzyme is a membrane protein, but it really has kind of 2 half components to it. One of these is actually quite soluble. So if we just kind of slice this enzyme right here, which actually is sort of what's done, we have this component up here, and this is where all the ATP synthesis is going to happen. In fact, not only is all the ATP synthesis going to happen here, it's only going to happen in these beta subunits. So each ATP synthase has 3 active sites, right? So 3 at a particular time, and the alphas are really just kind of going to be for spacers. And you can actually just kind of slice this thing off and add it to solution and make some ATP. You don't need any of the rest of this. So all this business with the proton gradient making ATP, that clearly can't be true, because if you just take these top pieces, either intact as it's shown, or alternatively break them apart and put them in solution with some ADP and some inorganic phosphate, you get ATP. That's absolutely the case. And so if we just kind of skip forward to this slide, we'll see something really, really weird. And this should really bother you because so much for biochemistry 1, throw up your hands. I guess that whole semester, just whatever. Here's the standard free energy of ATP synthesis in solution. This is the delta G naught that I told you about last semester, right? 30.5 kilojoules per mole. Kind of hard to measure it directly, but it's a very good accurate measurement. It's measured through coupling, but it is the correct number. If you add the ATP synthase, just those beta subunits now, to solution, and measure the standard free energy, you come up with 0 kilojoules per mole, and that is really bad for everything I told you about in biochemistry 1, because enzymes aren't supposed to change the equilibrium constant. So look, here we have an enzyme, and absolutely it has an active site, and it takes ADP, and it takes inorganic phosphate, and it brings them together, but the catalysis seems to happen by changing the equilibrium constant. I told you there was one enzyme that pulled this off, and this is the enzyme that nature chose to do it. First time it tried this strategy, last time it ever tried this strategy, this is an enzyme that actually shifts the equilibrium constant. This is the equilibrium constant when measured in solution, this is the equilibrium constant when measured. And this is really kind of bizarre, because enzymes are not supposed to change K. That's kind of not what enzymes are supposed to do. Any clues to that? And what are the significance? I mean, the first significance is, remember that whole thing about the proton synthesizing the ATP and the energy? I mean, yeah, probably not, right? Because you don't need the protons. You do not need them at all. You just put the beta subunit into your solution with ADP, inorganic phosphate, and you get some ATP. So what is the point of the whole rest of this? We just went through this whole very long discussion about the electron transport complex and protons and this side of the membrane and how much energy. Anyway, we do notice something really, really significant. You can measure the KD of ATP for both ADP and ATP. ADP binds probably as well as any substrate ever does. ATP binds really tightly. So what that means is that, yes, you can make ATP without protons. What's the problem? Why do you need the protons if it's not to make the ATP, really? What do do you need the proton's energy for? Look how tightly it's binding. That's like super tight. So what's the energy for? Most of it anyway. Yeah, kicking the darn ATP off. It binds so tightly to the ATP synthase, you need some energy to displace the ATP. Right? This is the consequence of using an enzyme to shift the equilibrium constant. So with that in mind, let's kind of take a look again at the ATP synthase and try to understand how this whole process is happening. First of all, of course, you have these 3 beta subunits that we're making 3 at a time. Alphas are really just here as spacers for our purpose. So each one of these is doing 3 ATP synthesis. If we kind of move down, we have this gamma subunit, which kind of looks like, I don't know, a cigar or something, a stick. It turns out that when we cut our way through this, which we will in a minute, we'll see that it's not quite symmetric. In fact, this gamma subunit is interacting differently with these 3 beta subunits. The consequences of gamma interacting differently with these 3 beta subunits is that even though they have the same polypeptide sequence, they have a slightly different structure. So gamma, because it's asymmetric, you really kind of can't see it from here, interacts differently with each one of these 3 different betas. And so even though they're all beta and they all have the same primary sequence, their structure is significantly enough different. If we go down here, so here's gamma, and then we have epsilon, which is connecting us into this membrane protein complex. It's just a proton channel, but it's a really weird proton channel. In fact, A here, the major component of the proton channel, is very bizarre because even though this is a proton channel, and proton channels allow protons to move from one side to the other, this is not a straightforward path. So imagine you're driving through a tunnel, and the tunnel entrance and exit are offset from each other. That's what A is like. You'll see that in a moment, right? You go drive into the tunnel, and you hit a wall, and then the other part of the tunnel, I know, someone in New York City would probably build something like this. This is not shocking. I ride the subway every day out there. But it's an offset proton channel. It turns out that the only way to make it from proton, to make it from one side to another, is to actually enter into one of these C subunits. Now notice that I say C or C10. C can be 8, 9, 10, 11, 12, 13, 14, possibly fewer, potentially more. It depends on what type of cell you're in, depends what type of organism you are, and it may turn out to be quite stochastic anyway. There's no fixed number. C of 10 just is fine here. There are these 10, and it turns out that a proton will need to move through each one of these 10 different subunits before it can finally leave to the opposite side of the membrane. So the pathway for a proton to move, you can see here, it'll enter into A, it'll hop onto the first C subunit. When it hops onto the first C subunit, that one moves like a carousel out of the way and leaves another empty one here, and then the proton moves here. So it has to go from proton to proton to proton to proton, ride its way, a single proton has to ride all the way through the carousel before it gets to the second half of the tunnel and can actually leave. So these protons are actually moving the wheel around. Just to be clear, it's not you're hopping from one C to the next. It hops on, and then the whole C subunit moves. And the reason why it does so is that right here at the entry point between A and the C subunits, there's an aspartic acid. That aspartic acid is deprotonated. And so it's actually reaching out of the membrane. That aspartic acid is pointing straight up out of the membrane. But when it picks up the proton, it becomes, it becomes soluble in the membrane, it becomes more hydrophobic. And so that aspartic acid lunges down into the membrane. And that lunging down motion actually pushes the C subunit forward. So you grab a proton and lunge. And now the next one behind you grabs one and lunges, and it pushes that C subunit out of the way. And then the third one grabs a proton and lunges and pushes it out of the way. And so this C subunit just keeps moving, being pushed by the ones before it until it makes a complete turn. Once the last one goes there, there's another amino acid here to accept that proton, and it flies out of the membrane. So the only way the proton can get from one side of the membrane is to hop onto a C subunit and then ride this carousel all the way around, pushed by the subunit behind it, until finally there's this exit part and it comes out. What's the point of all this moving? All this moving is connected to these epsilon and delta subunits here. That's causing this stalk here that was pushing into the betas to rotate, to spin. And in fact, you can remove the alphas and betas, and that central stalk is really just spinning around. Why is that central stalk here, this one just here, spinning around? Now remember, because it is asymmetrically shaped, as it spins, it's changing the conformation of each of these beta subunits. This starts to explain what's actually going on here. So here's now a cross section through. Here's the alphas. They're not doing anything. They're just air spacers. Here's our 3 beta subunits. 1, 2, 3 beta subunits, differently colored because they are in different conformations. And now you can see that gamma subunit is asymmetric. So you'll notice that each of these beta subunits in primary sequence have a slightly different shape. These different shape structures have a different affinity for ATP and ADP. And so we call this, how this actually results in the synthesis of ATP, the binding change model. So here we could look at just follow one beta subunit through. Here in this bottom we're following 3. So we're always making 3 ATP at a time, but we're at different stages of production. So let's just look at one. We start here in the open. And the open subunit here is a weak affinity but decent for ADP and inorganic phosphate. So they bind, a proton comes in and takes us to loose. We now increase our affinity for ADP. This is a conformation that has an increased affinity for ADP and inorganic phosphate, so it cannot leave. Another proton comes in, takes us into tense. Our affinity for ADP is low, our affinity for ATP is higher. So as ADP and inorganic phosphate are next to each other right now, suddenly the structure changed from one that has a high affinity for ADP, a low affinity for ATP, to one that has a high affinity for ATP and a low affinity for ADP, so ATP is bound. Formed. That's it. That's would have happened again, even if there was no proton gradient, this would have occurred, right? We kind of already saw that. But now we have a problem, because this thing has a ridiculously high affinity for ATP. And so the proton comes in, shoves us back into this conformation, which has almost no affinity for ATP at all, and in fact has a higher affinity for ADP. And you can see that we are making 3 of these at a time, because as this gamma subunit is being spun by those protons, because it's connected to those C subunits, right? It's going through the different stages of ATP production. 3 being made at a time, but not in the same stages of production. This again, all collectively again, is what we know as the binding change model. So here we go, right? We have ADP and ADP present. Let's just focus on one particular subunit here. ADP is loosely bound. Protons are coming in, hopping onto the C. That causes them to lunge forward when they pick up that aspartic acid, move a new empty C behind them. So the C moves forward by 1. Then another proton, C moves forward by another. When they moved forward, this went from having a low affinity of ADP to a high affinity for ADP and inorganic phosphate, so they're not leaving. Another proton came in, changed the affinity of that particular subunit to one that has a high affinity for ATP, low affinity for ADP. ADP, and inorganic phosphate were next to each other. ATP got formed, but again, we needed another one to come in, right, to just knock that off. And so this process is continuously spinning this wheel, changing these beta subunits between these various different conformations, and that's what's allowing the ATP to get synthesized, the ADP to bind to get synthesized, and then for the ATP to get released. So the letter J really is C. So we can kind of see just schematically how the process is actually happening. The proton flow through, causing the wheel to spin, causing the changes in the subunit composition of that beta subunit. A couple of important things then. Now that we know how the ATP gets made, it's time to pay the price, right? Because we, of course, created a proton gradient. That's great. We can make ATP. But we need raw materials to do so. We've kind of ignored that. And unfortunately, the import of these raw materials is going to cost us a little bit of energy. What do I mean by it's going to cost us a little bit of energy? Well, what do you need if you want to have ATP synthesized inside the matrix? You need phosphate and you need ADP. And bringing both of those in is going to take some energy and therefore lower the amount of energy that we actually have, at least in theory, for making ATP. Let's look at the inorganic phosphate first. Inorganic phosphate turns out to enter by a symporter. It symports in with protons, right? So some of the proton gradient that you created is not going to be used directly to make ATP. Some of those protons that were still logically pumped out of the membrane are going to come back in to bring the inorganic phosphate in. It is with a symporter. Electrically, it's not changing anything. This totally is neutral, but it is depleting your osmotic number. What about the ADP? How about that? It turns out that ADP and ATP exchange each other through an antiporter. So there's really not an osmotic situation. I mean, you're losing But look at the charge in which they're at these various pHs. You're sending a -4 charge to this side of the membrane to bring in a -3 charge on this side of the membrane. Now again, you're not affecting the osmotic state of things, but you're moving more negative charge to this side than you are to this side, so that's also lowering the total amount of energy. You're essentially putting a net minus charge, where you want there to be a positive charge. So some of the energy is actually lost in this actual translocation step, but that's a necessity, right? You can't make ATP if you don't have inorganic phosphate and inorganic phosphate ADP. What about those electrons that came from glycolysis, right? Notice that I was very, very specific in talking about NADH from the citric acid cycle, again, not from glycolysis. Since we're talking about moving things across the membrane, what about those? We know that there is no way to move NAD or NADH across a membrane. So we've already seen that we have to pay the price for moving ADP and inorganic phosphate. What about those NADHs from glycolysis? We cannot just take NADH and push it across the membrane, and this creates a problem. Not a problem for the NADHs, by the way, that were there in the citric acid cycle, because those were actually made in the matrix, but they're just hanging out in the cytosol, and we'd like to use them. It turns out that we have two ways to move those across, and this is another piece of evidence that nobody thought about this, because we'll see that one of those ways preserves the energy, the other way dissipates the energy. The energy wasteful form is in the two most metabolically active tissues in your body, the skeletal muscle and the brain. So if you're doing this on the test and trying to recall between the two different mechanisms, just remember, no, nature got it backwards. There's a very energy efficient way to move those electrons across, and a very energy inefficient way to move those electrons across, and your skeletal muscle and brain, the two organs that need energy the most, turned out to use the less efficient pathway. So proof once again that nobody designed this. Anyway, let's look at the first way. This is in skeletal muscle and brain. Again, our problem is that we have NADH electrons out here in the cytosol. No way to move NAD across the membrane. We did see that we pay a cost for transporting in. Here's the cost we're going to pay for moving those NADH electrons. What are we going to do? We're going to siphon a little bit of molecule out of glycolysis, the dihydroxyacetone phosphate. This is not where the cost is, by the way, because that DHAP can later go back into glycolysis. So this is not what I mean by the cost. We're going to dump the electrons onto them to make glycerol 3-phosphate. Then we are going to take the glycerol 3-phosphate and oxidize it back to dihydroxyacetone phosphate. So again, nothing was lost from glycolysis in here, but we move them across the membrane. Hooray! Why is this so unideal? Why is this so inefficient? Well, we move them from NADH, right, to dihydroxyacetone phosphate to make glycerol 3-phosphate. That's fine, but the enzyme that oxidizes them uses FAD. What does that mean? It means that we can only transfer these to Q to make QH2. We're not transferring them to NADH, so they bypass complex 1. So NADHs that are generated by the citric acid cycle can pump 10 protons. NADHs that were generated in skeletal muscle and the brain by glycolysis can only pump 6 protons. We just skipped complex 1. That was a loss. They were NADHs, but they're not the same. So this is what we call, again, the glyceraldehyde phosphate shuttle. Same thing. I just really like both slides. They're kind of indecisive. So it's not two different processes, just to be clear. What about every other tissue? Every other tissue uses what seems like a much more complicated process, but it turns out to be much, much more efficient. So this is the malate shuttle. You're going to see some proteins that you've already seen before. That's a good thing. There's going to be some antiporters here. It looks really complicated, but what we can do is break this down into the outer ring and the inner ring, and then just kind of, it will make much more sense, I promise. Let's worry about the electrons first, okay? The outer ring is predominantly from the motion of electrons, so this is the ring that accomplishes our goal. We have oxaloacetate in this cytosol. We've already seen that before. We're going to dump the electrons onto them to make malate. We know oxaloacetate cannot enter into the mitochondria, but we know that malate can. We've seen that happen quite a number of times already. Once we get into the mitochondria, we're just going to reoxidize that malate back to oxaloacetate. Why? Well, that's great, right? Because we moved an NADH and yet across the membrane without having to expend anything. The issue here, of course, is that now we have all this oxaloacetate on this side of the mitochondria. That might be a good thing, but really we're risking depleting everything out here of carbon. So we need to get that oxaloacetate back out to the cytosol, so we can use it again, so we don't have to take all of our oxaloacetate. Unfortunately, we already know there's no way to move oxaloacetate across membranes. We've said that so many times that it doesn't bear repeating. But we do know from other processes that if we transaminate that oxaloacetate, we can send it out in the form of aspartate, and then the aspartate can leave and reverse the transamination back into oxaloacetate. That's the outer ring. That's it. And if you ask why this is beautiful, it's because we net moved an NADH from this side of the membrane to this side. We don't bypass complex 1, which means this one can pump 10 protons. What is the problem with just having the outer ring is that quite quickly we're going to establish, of course, a carbon and nitrogen imbalance unless we keep everything going. So we want to make sure that things are balanced. How do we make sure that things are balanced? Well, first of all, we're going to have some antiporters. We're going to make sure that anything that leaves, one comes back in. Who's going to antiport? Well, our concern here, right, is our nitrogen. We need to keep our nitrogen cycle. We have glutamate, and the glutamate is going to be doing our transaminations. But if we don't have somebody bringing the glutamate in, we're going to quickly deplete that. And so we antiport the aspartate with the glutamate. The aspartate goes out. When glutamate comes in, it donates its nitrogen to the transamination process. Of course, that's going to deplete the carbon, right? We're going to end up with a whole bunch of alpha-ketoglutarate here, but that's okay because the malate is going to turn out to operate in antiport with the alpha-ketoglutarate. So the outer ring is primarily for the transport of electrons. The inner ring is to make sure that we keep the carbons and the nitrogens balanced so that we're not depleting one of these preferentially over the other on the opposite side of the membrane. This is the malate shuttle. This is the best way to do it. Nothing is lost. NADHs from the malate shuttle that came from the cytosol are just as good as citric acid cycle NADHs. Unfortunately, your brain and muscle, the two most metabolically organs, don't use these. So that is just what happened in nature. Anyway, here's the malate aspartate shuttle again, just showing to you a little bit more obviously clear perspective. The outer ring, remember, is your electron ring, right? This is the one that's just moving electrons in. Basically reduce it, oxidize it, send it back. These other rings here, what we call the inner ring and outer ring and the other inner ring and the other perspective, these are just for making sure that we're maintaining balance with carbons and nitrogens so that no one side of the membrane is preferentially getting depleted or too many carbons or too many nitrogens. Anyway, time to keep track. Let's do some scoring. You have probably seen, first of all, if you saw this in a bio textbook, a little squiggle. The reason why there's that squiggle is that it turns out that there's actually no way to actually know the number. When your bio textbook said 30 to 34 or 32 to 34 ATP, that's because that C number can even be variable within a cell and within an organism. So it's not clear that that's a stoichiometric thing. That being said, if you were to do this on a test, which is a decent chance you're going to have to do, I don't want to see squiggles. I want to see absolute calculations. So I'm going to give you a defined C number. And here the C number is 12. So let's see how it is that we get up to this. Let's say exactly 32 ATP per glucose. How do we do that? First of all, we're just counting ATPs that are actually directly generated. We have two processes. Glycolysis per glucose is going to net us 2, right? Citric acid cycle 2 times around, 2 nucleotide triphosphates. I'm assigning them both ATP, but you could just call them NTPs, and that's perfectly fine. So that's coming from our substrate level phosphorylation. So let's just shove those 4 aside. They're good, but let's put them aside. Now we have 2 NADHs. You notice I'm saying 2.5 ATP per NADH, so I'm using the efficient shuttle here. But let's not start counting ATPs yet. Let's start counting protons together, okay? Because this is what you're going to have to do if I change that C number, so we might as well do so. So 2 NADHs, I'm going to count these as 2 NADHs, 10 protons per run through, right? 20 protons. Again, using the efficient shuttle. I could give you the non-efficient shuttle as well. 20 protons. Here's another 20 protons. Another 20 protons are coming from here. 2 NADHs, 10 protons per NADH because we're using complex 1, 3, and 4. So we're up to 20 protons. I'm really not that good at doing this on my own, so let's hope I get this straight. Now we have 6 20, so that's 80 protons, and this is 2 FADH2s, right? So that is 6. So what do we have here? So many count my protons here, 20, 40, 60, 70, 80, 90, 100, 112 protons? Is that right? Is that yes? Yes? Okay, so we netted ourselves 112 protons total, again, using the most efficient shuttle. C is equal to 12. So that means that we need 12 protons to synthesize 3 ATP, right? So if we take 112 protons, 3 ATP per 12 protons, that's what C is equal to 12 means. 12 means you take 3 ATP protons. I believe if you then bring in those 4 ATPs that we shuttled out of glycolysis, you come up with exactly 32 ATP per glucose. Challenge me if I'm wrong, people. I'm perfectly happy to be, but that's as good as the math as I can possibly do in my head. That's where you come up with this. So what can change in this problem? I mean, a couple things, right? C is not necessarily 12. C can be anything from 8, I believe this highest C that's ever been measured is 14. The more higher C is, right, the more protons it takes to synthesize 3 ATP. The other thing is I used here, this is my little shorthand to remind me, 2.5 ATP for NADH. That meant I must have used the malate shuttle, but I could have used the glycerol phosphate shuttle. That was going to bring me down to, instead of 20 protons here, actually only 12 protons. So does anybody not understand the calculation we just did? I mean, it's kind of worked out in the notes, but let's make sure, there's only a few more slides after this, so let's make sure that we understood what we did. 20 protons, right? 2 NADHs, 20 protons there. 2 NADHs, 20 protons, right? Again, using the efficient shuttle mechanism, and this is already the mitochondria. So that's 40 protons, that's another 60 protons. 60 plus 40 is 100 protons from NADH. FADH2s bypass complex 1, so we only got 6 protons per, so that's 12. Right, so that is 112 protons were generated for each glucose molecule. 112 protons for 12 protons for 3 ATP, netted 28 ATP, and then we also had 4 ATP, 2 of which were synthesized in glycolysis, 2 of which were then synthesized in the citric acid cycle. That's how I came up with 32 ATP. So no memorizing numbers for the test because it's very unlikely I give you C is equal to 12 now with the malate shuttle if that's an actual test question, right? You want to make sure that you actually know how to work through this. Just the last thing today, and then we just have like 3 slides so we can pick that up tomorrow. There are some cells that have a slightly different way of doing this, and these cells are called brown fat cells. You don't have them anymore, but there are 2 really adorable creatures on Earth that do. 1 are babies, and 2 are hibernating bears. I know. Adorable for different reasons, right? Both of those have the issue that they are not metabolically active sufficiently to generate enough body heat to keep from essentially freezing and dying. And so in brown fat, you actually have this protein here called uncoupler protein that effectively works like 2,4-dimetrophenol. What happens? You are metabolizing energy, right? You are generating a proton gradient, but instead of going through an ATP synthase, it just goes through this proton channel. Because it goes downhill, it releases energy, but because there is no wheel to move, no ATP to synthesize, all that energy is just lost in heat. And so infants do this because they are not yet metabolically have enough tissue to sustain a body temperature, newborns I should say, and hibernating animals do this. And if you've heard that bears generate their own water during hibernation, they sure do. Here's where it's coming from. It's coming from their brown fat tissue in this process of this respiratory chain, right? They just don't use an ATP synthase in these cells. They instead have this uncoupler protein here. Great, so we'll pick this up just a couple slides next time, and then we're going to see how we get other types of molecules into the electron transport.